Hello and welcome to Oro-Oto Cerebral Pathway channel. In the embryology series, today we are going to talk about gastrulation. Gastrulation is all about getting trilaminar. That is, when a bilaminar germ disc gets converted into a trilaminar germ disc, it happens during the third week of intrauterine life, and gastrulation is all about conversion of this bilaminar disc into a trilaminar one. The important event which happens during gastrulation is the three definitive germ layers are formed, and in addition to that, the axial orientation of the embryo is also established. And this interesting stage of gastrulation begins with formation of a significant structure called the primitive streak. So let us directly jump in. The primitive streak appears caudally in the median plane of dorsal aspect of the embryonic disc. Cells of the epiblast proliferate and migrate to the median plane of the disc to form primitive streak. And this proliferation plus migration involves another significant event, the epithelio-mesenchymal transformation. Remember, bilaminar germ disc consists of two layers, epiblast and hypoblast. Now, since epiblast is a layer, the physical properties of the cells of epiblast are going to be much like epithelial cells, where they come together and form continuous sheet or a layer. Now, for the cells of the epiblast to be able to transform themselves and migrate, they need to shed off their epithelial properties and be able to move about from place to place. That will be possible only by transforming themselves into a mesenchymal cell. So this is what epithelio-mesenchymal transformation is about. Early in the initial part of third week, the primitive streak is vaguely defined. But by the day 15 to 16, it is clearly visible as a narrow groove with a slight bulge on each side. This groove is termed as primitive groove. At the cephalic end of the groove, there is a slightly elevated area, the primitive node, which is also called primitive knot or the Henson's node. At the center of the node, there is a depression. This depression is the primitive pit or the blastopore. After this epithelio-mesenchymal transformation, cells of the epiblast migrate to the primitive streak, proliferate, and on reaching the primitive streak, they become flask-shaped, detach from the epiblast, and slip in the interval between the epiblast and the hypoblast. This migration is described as a process of invagination. As more and more cells migrate, they come to occupy the interval between the epiblast above and the hypoblast below. It can rather be said that they create an interval between the epiblast and the hypoblast and occupy that interval. As gastrulation progresses, the fate of the invaginating cells depends on the location at which they migrate. Some of the cells which are present in the hypoblastic domain displace the hypoblast cells and get converted into endoderm, or they form the endoderm. The others that lie between epiblast and the endoderm will transform into mesoderm, which can now be called as intra-embryonic mesoderm. So the earlier mesoderm that was formed in week 2 was extra-embryonic mesoderm, and the new mesoderm that is formed in week 3 as a result of gastrulation is the intra-embryonic mesoderm or the definitive mesoderm. And in addition to these two, the other cells as a result of gastrulation which remain in the region of epiblast or stay in the epiblastic domain, they will transform themselves into ectoderm. And that is how, by the process of gastrulation, the epiblast helps in formation of the three definitive germ layers, the endoderm, mesoderm, and ectoderm. So it won't be wrong to say that the epiblast is the source for all the three germ layers. It is a mother for all the three layers. So now we have a definitive endoderm, a definitive mesoderm, and a definitive ectoderm. That can also be said as we have intra-embryonic ectoderm, intra-embryonic mesoderm, intra-embryonic endoderm. But this term intra-embryonic is mainly used in connection with the mesoderm. Right. Cells migrate laterally and cranially. We are talking about intraembryonic mesoderm, where the cells migrate laterally and cranially. They migrate laterally and establish contact with the extraembryonic mesoderm, and cranially they pass on either sides of the precaudal plate. I'm sure you remember the precaudal plate. It's a circumscribed area towards the head end of the embryo where the epiblast and hypoblast are tightly attached or fused to each other. So what are the areas where the ectoderm and endoderm are tightly attached to each other? Well, cranially, in the region of precaudal plate, we have the tight attachment between the two. Now this region of precaudal plate in future is going to give rise to the buccopharyngeal membrane. In the caudal region of the embryo, caudal to the primitive streak, we have yet another area where the endoderm and ectoderm are tightly adhered together. That region is the cloacal membrane. And there is one more region where the intraembryonic mesoderm itself transforms into a prenotocaudal process or the notocaudal process. So that region is the one which is going to form the notochord. This region that is supposed to form notochord will be lying in the midline. So these three regions, buccopharyngeal membrane region, the cloacal membrane region, and the region of notochord, are the areas where the mesoderm does not extend into the space between ectoderm and endoderm. In the cranial region, cranial to buccopharyngeal membrane, the intraembryonic mesoderm of both the sides is in continuation with the migration of the opposite side. And this area, a horseshoe-shaped area present around the cranial end, which surrounds the buccopharyngeal membrane, that part is the one which gives rise to the heart tube and hence will now be called as cardiogenic area. In future, during embryonic life, the buccopharyngeal membrane breaks down to form opening of the oral cavity, and the cloacal membrane also breaks down to form the opening of the anorectal and the urogenital tracts. So what is the fate of this primitive streak? Well, the primitive streak was formed to give rise, it was a mechanism for the epiblast to be able to form the three definitive germ layers, and once that is done, the primitive streak disappears completely by the end of 4th week. After formation of the intraembryonic mesoderm, the primitive streak starts regressing and it completely disappears by the end of 4th week. Well, sometimes the primitive streak does not disappear altogether and some parts, some cell clusters of it remain, and when they do, they give rise to a tumor called a sacrococcygeal teratoma. Well, this, going by the name, should be present towards the caudal side of the embryonic body. In addition to that, abnormal gastrulation may lead to caudal dysplasia or caudal agenesis. The severest form in this series is a sirenomelia or mermaid-like habitus. Well, this is all about the gastrulation that you are supposed to know in brief. Happy learning!